Hello, my name is Michelle Finger, and I will present my human disease pedigree. For my presentation, I chose the G6PD deficiency, and the G6PD is an enzyme called glucose 6-phosphate dehydrogenase, and it has the function of reducing ribose 5-phosphate in the generation of NADPH in the hexose monophosphate pathway. So in other words, it protects the erythrocyte from oxidant stress and premature lysis. The G6PD gene is located on the chromosome X, specifically in the XQ28 location that is showed in this picture right here. The inheritance pattern of this disease is a recessive X-linked inherited disease. And one thing I have to pinpoint, and the reason why this happens, is that during the embryonic development in females, there's a response to the genomic imbalance of the mutation of G6PD, which causes one chromosome X inactivation in most females. This causes women to be heterozygous recessive or carriers. Males with a single X chromosome can either have a normal G6PD genotypic levels, unaffected, or deficient or affected, or they exhibit the phenotype for this disease. In a few cases, heterozygous females carry one allele encoding a normal G6PD enzyme that functions normally, or they encode another allele with G6PD deficient function. Since there is a mechanism of random X chromosome inactivation and double X alleles in women, there is a certain buffer effect in this gene that will protect, in most cases, women from expressing this genotype. There's rare cases in which unfavorable X inactivation could happen, where the unaffected X chromosome is silenced in most of the cells, such as homozygous females, and they can present the phenotype symptoms and all the triggering factors that come along with this disease, because they have a random mutation in both X chromosomes. Another reason why women can have the X-linked recessive disease is because the father is affected and the mother is a carrier or affected. Daughters of female carriers of the X-linked G6PD disorder have a 50% probability to be carriers themselves, and boys have 50% to be affected. And males with X-linked disease can pass the gene to all his daughters, who will be carriers if their mother is unaffected. And males cannot pass the disease to his sons because males will always pass their Y chromosome instead of the X chromosome to all his male offsprings. As depicted in my pedigree drawing, I will first talk about the first, second, and third generations. The first generation has a marriage between male 1 and female 2, which is a case of a carrier female and a healthy male. They had 3 offsprings, which is female 2, deceased female 3, and affected male 4. Following my Punnett squares on my left side, there's a 50% probability of healthy females that wouldn't express the phenotype, which is the case of female 2 and deceased female 3. Males have a 25% to be affected, and such is the case of male 4. Going to the second generation, healthy female 2 marries affected male 1. Then they had female offsprings number 1 and 3. Both will be carriers. And healthy male offspring number 2. In this case, the father of the second generation expressed the phenotype of G6PD deficiency, and all his female offsprings have a 50% chance of being carriers themselves because he's passing his only mutated X chromosome to his daughter's chromosomes. The same situation is happening with affected male 4 and healthy female 5 from the second generation. All female offspring, such as female 5 from the third generation, is a carrier, whereas the male offspring number 6 is healthy. My final example of this family tree is the third generation, where male 6, which is healthy, has children with female 7, which is the rare case that I explained earlier of a homozygous G6PD female. Under this rare circumstance, the likelihood of most of their offspring getting the genotype for G6PD deficiency is high. Like a 50% probabilities of female offspring will be carriers, and a 50% of males are going to be affected in the fourth generation. Offspring number 9 was an affected deceased male that passed away because of brain damage caused by a hemolytic crisis that was not treated, and among other complications. I will explain further on why something like this could have happened. The G6PD is found in the cytoplasm of all cells in the body, and it protects the cells from oxidative stress reactive oxygen species called ROS. Erythrocytes are particularly vulnerable to these RS agents due to the function of oxygen transportation and the inability to replace cellular proteins as mature cells. The inheritance of G6PD deficiency may result in acute hemolytic anemia through the increases of ROS. This could be triggered by the exposure to certain foods and medications that I will explain later on. And if people with G6PD deficiency are triggered by these RS agents, they will have the following symptoms, which it will be jaundice, yellow skin, fatigue, paleness, rapid heart rate, enlarged spleen, shortness of breath, and hematuria, which is blood in the urine. G6PD deficiency is like anemia, but it can develop very quickly, leading to an increase in the symptoms mentioned. Infants actually can get severely affected by G6PD deficiency, and they can develop a symptom called severe jaundice, which can appear in the infant during the first 24 hours. And if it's left untreated, these infants can develop brain damage among other complications, which is the case that I mentioned for deceased male, affected male number 9. In many, many cases, people may not experience signs or symptoms at all, or they're actually unaware. How is G6PD deficiency diagnosed? So first of all, there will be a check of the complete medical history and verification of family G6PD records. Doctors may test first for hemolytic anemia because the symptoms are very, very similar to hemolytic anemia. And the tests for erythrocytes will be lactate dehydrogenase, bilirubin levels, complete blood count, reticulocyte count, serum aminotransferases, and peripheral blood smear. There are some specific tests that are for G6PD levels that are quantitative and qualitative as well. And about the treatment, the G6PD deficiency can be cured because it's a genetic disorder. However, some substances should be avoided to prevent the symptomatic chain of events from G6PD deficiency. Some contraindications that literally means that people can't take these substances, otherwise they will have very bad symptoms, are medications such as anti-malarial drugs, primaquine, vitamin K, several antibiotics such as streptomycin, and NSAID aspirin. Foods like fava beans that are shown in the right side, and many other medications should be avoided, but the list is so big I cannot add it here. But the references found in this citation right here. There are extra recommendations to have a better quality of life for the G6PD deficient individuals. That it doesn't necessarily mean that if they consume those substances, they're gonna have very bad reactions. It's just certain things that are considered and should be avoided, such as big quantities of alcohol, smoking, high intensity workout can trigger high oxidative levels, stress, and sleeplessness. Some people and newborn babies have more severe symptoms. And if there's a hemolytic anemia crisis, patients may need a blood transfusion. And for newborn babies with jaundice, healthcare providers may treat it with phototherapy, which is a natural artificial light treatment. And if there are serious cases, there could be an exchange of blood transfusion of new donated blood if it's necessary. Here are my references from all the information I just mentioned, and the image references as well. Thank you very much.